Everyone, now buckle up for a dive into another microbial world. Today we are exploring the genus Serratia, Erwinia and Yersinia, three fascinating members of Enterobacteriaceae with surprising diversity. These guys are from hospital pathogens to plant pathogens, so get ready to discover their unique characteristics and other features. I am your regular tutor, Bamidele Odumosu. Let's go. Serratia are facultative anaerobic bacteria, just like every other members of Enterobacteriaceae. Facultative anaerobic means they can survive with or without oxygen. They are widely found in the environment. Some species of Serratia can act as opportunistic pathogens, causing infection in people with weakened immune system, immunocompromised system, or those who are in healthcare settings. Now, we have one important member of genus Serratia, which is Serratia macensis. It is the most common of human infection, occasionally found in the environment, producing a red pigment called prodigiosin. Serratia macensis is responsible for hospital-acquired infections like pneumonia, urinary tract infections, and wound infection. We also have another member, which is Serratia plymoutica, which is less common among those causing infection, or it can be found in the water and soil. We also have leucophacian, which rarely cause infection, but mainly associated with hospital environment. Rubidae are those that cause infection, mainly reported in immunocompromised individual as well. Then we have odorifera, which also rarely cause infection, but is known for its characteristic fruity odor. We have other species of Serratia that exist, but their role in human disease are less known, so they don't have much relevance, unlike the main Serratia macensis, which is the one that is mostly implicated in infection. So, Serratia macensis is the normal pathogen that is usually associated with nosocomial infection. I believe we all know what is nosocomial infection. These are hospital-acquired infections that occur 2 or 3 days after a patient has been admitted in the hospital. These come with different type of disease, not what the patient came in with. So their normal habitats include water, plant, soil, animal, household item, hospitalized patients, virtually everywhere. So like I said, they produce red pigment called prodigiosin. The prodigiosin in Serratia is a red antibiotic pigment produced by some strains of Serratia, especially Serratia macensis. It is a very stable pigment which usually resistant to light, heat, and even to some disinfectants. This is responsible for staining on surfaces like bathroom tiles, bathtub, contact lenses, and it also cause some pink stain on some devices. Serratia can cause diverse infection, which include pneumonia, UTI, and wound infection, like I said, and they are opportunistic pathogen, and usually they infect people with medical implants and immunocompromised patients. Typically, they grow with a red coloration in the laboratory, so they appear on a blood agar as a red color in laboratory identification of Serratia. Their appearance on Maconche is a dry lactose fermenting colony, which shows that they can actually ferment lactose, but they are not heavy lactose fermenter. They are motile with flagella, and they are citrate positive, and they produce pigment, which is the red pigment that is characteristic of Serratia. Serratia can be distinguished from other family of Enterobacteriaceae because of their ability to produce DNase. This is an enzyme that helps Serratia to break down host cell DNA for nutrients and aids its invasion. It also assists or facilitates its biofilm formation, which also enables it to resist antibiotic resistance. The DNase also help the Serratia to digest extracellular DNA, which provides scaffold for their biofilm formation. So not only DNA, they also produce another type of enzyme, which is known as lipase. Lipase helps Serratia to break down fat and oil, which they can also use as energy source. This also is a virtue because not all microorganisms have ability to produce lipase. So this helps this pathogen or opportunity pathogen to invade the host tissue and help it to penetrate into deeper sites. So the lipase may be playing a role in a virulent factor for Serratia because normally oil, fatty acids can provide a shield in the cell, whereas ability to break down this fat and oil is a virulent factor that can allow the Serratia to penetrate to deeper tissue of the host. The third enzyme that is produced as a differentiation from other Enterobacteriaceae is the gelatinase. Gelatin is a protein that is present in several tissues, including the bones, skin, and connective tissues of the body. So, gelatinase allow machines to break down the tissue, thereby contributing to tissue damage and spread of the organism in the host cell, thereby spreading the infection. So these enzymes, DNase, lipase, and gelatinase, are factors present in Serratia that enables its virulence in the host system. Economic importance. They are usually associated with nosocomial infection and they cause various disease and infection in humans, especially urinary tract infection and pneumonia. It should be known that Serratia is an opportunistic pathogen with its ability to penetrate deep into the host tissue. It also possesses virulent factors as discussed. Hence, Serratia is a notable member of Enterobacteriaceae as we have discussed. So we are going to look at another member of Enterobacteriaceae, which is genus Erwinia. Erwinia is a genus among the family of Enterobacteriaceae that primarily affect plants. They are plant pathogens. They cause a variety of diseases. They are named after Erwin Frank Smith, a famous plant pathologist who discovered this plant pathogen. They causes fire blights, they cause apple rots, soft rots, pears, potato destruction, carrots, other vegetables belonging to Rosaceae crop. So they affect all kinds of plants disease. So they cause bacterial weed which affect all these plants. So a common member of this genus is Erwinia amylovora. which infect woody plants and other susceptible plants. It is a typical plant pathogen. Erwinia amylovora exhibits cyclotolerance, enabling it to survive and proliferate at comparatively low temperature. These characteristics facilitate its persistence throughout the winter season, positioning it to prompt infection early with the subsequent growing season. The bacterium inhabits the mouthpart of insects, serving as a vector for dissemination to other flowering plants, hence causing infection. How do we identify a winner in the laboratory? Because it is a primary soil habitat. So this necessitates selective media for Erwinia isolation. And due to their predominant presence in soil, Erwinia bacteria demands specialized media to suppress the overwhelming growth of other soil-dwelling organisms. The soil is the hub of several varieties of microorganisms that grows and compete with one another. So in order to suppress their growth and promote the growth of Erwinia, several effective selective media can be used, which includes crystal violet bile agar, CBBA, Seagull's antibiotic eosin blue medium. We also have Suyama and Sakamoto's modified Vygotsky medium. These mediums are selective and they allow Erwinia to grow. The identification of Erwinia relies on the distinct growth and morphological characteristics exhibited on this individual selective agar based on Erwinia identity. Hence, Erwinia can be isolated. Erwinia is an economic important pathogen associated with disease in plants. Like I said earlier, they are associated with different types of infection. So they pose a significant threat to agriculture, particularly in regions where herbaceous fruit like apple, pear are grown, thereby causing losses of harvest during harvest time. Erwinia is the only member of Enterobacteriaceae that is widely known that is associated with plants as a pathogen, and it is not usually found causing disease infection in humans. Majority of the known Enterobacteriaceae we have discussed are usually associated with nosocomial infection or community acquired infection. So we will look at the third member of Enterobacteriaceae that we are looking at today, which is the genus Yersinia. Is an interesting genus. It consists of bacteria with diverse roles. Yersinia itself is a bacteria belonging to the family Yersiniaceae. Now, Yersiniaceae is a new family carved out of Enterobacteriaceae. So Yersinia is no longer considered as a family of Enterobacteriaceae. While many sources may still consider Yersinia as a member of Enterobacteriaceae, the current taxonomic classification places Yersinia in its own family, Yersiniaceae, in 2016. And this is based on extensive genomic analysis that reveals significant difference between Yersinia and other family of Enterobacteriaceae. That is the wonderful thing about science. It keeps evolving The use of high-throughput sequencing and genomic analysis has allowed more differentiation of bacteria that were previously lumped together as belonging to the same family, now on a separate basis. So, we are still considering Yersinia as a member of the family closely related to Enterobacteriaceae. So, we are going to look at these properties of Yersinia. Yersinia are easily found in the environment. They are found in a variety of environments, which include soil, water, and in the intestine of some animals. While some species of Yersinia are harmless, others are known to cause a variety of diseases in human and animals. They are slow-growing, non-motile, facultative anaerobic member of the family Yersiniaceae. They appear in plump cucurbacilli. Sometimes they appear in pairs or single cell. So, it is not all the members of Yersinia that are pathogenic. However, there are species of Yersinia that are pathogenic in humans. This includes Yersinia pestis. Yersinia pestis is the most famous member of this genus and responsible for causing the deadly plague. The plague has swept across the world in multiple pandemics throughout history, causing immense suffering and death to individuals. So it is the major cause of this bubonic plague. Plague is a zoonotic disease transmitted from animals and their infected fleas. It is usually associated with rodents. Yersinia pestis primarily infect rodents, but it can be transmitted to humans through the bite of infected fleas. The symptoms include plague, fever, chills. Symptoms of plague include chills, fever, and swollen lymph nodes, and sometimes, in some severe cases, it could have septicemic form or appear in pneumonic form. So Yersinia pestis is one important member of this family. Yes. Then we also have another member, which is Yersinia enterolytica. Yersinia enterolytica is a common cause of foodborne illness in humans, and this can be known as Yersiniasis. Yersinia enterolytica is typically transmitted through contaminated pork products, contaminated water, and other contaminated cattle products, and unwashed vegetables. It also causes yersinosis, which has a similar symptoms of fever, abdominal pain, and diarrhea. Then we have Yersinia pseudotuberculosis. This species can cause a similar illness like yersinosis, but it is less common. It is less common and not really encountered. However, whenever it is encountered, it is usually transmitted through contact with infected animals or contaminated food. The symptoms is also similar to enterolytica in terms of yersinosis, such as the fever, abdominal pain, and diarrhea. In isolation of Yersinia, Yersinia consists of fastidious species, although pestis is not really fastidious. But yes, Yersinia enterolytica and pseudotuberculosis are actually more fastidious. Hence, they can be very difficult to grow, which means they require enriched agar containing more nutrients. So, example of enriched agar is blood agar. Now, the blood agar, especially defibrinated horse blood agar, which allows them to grow and suppress other organisms. They are slow-growing bacteria, and they can also grow in brain-heart infusion broth. They can also grow in selective agar such as maconche or eosin methylene blue agar. So because of their slow-growing nature and fastidious growth condition, Yersinia can be cultivated at the optimal temperature of 28 to 30 degrees and ambient temperature atmospheric oxygen of 5% carbon dioxide acceptable, and the incubation is up to 5 days because they are slow growing organisms. That is why they have to be grown on specialized agar to prevent other fast growing organisms from overcrowding the plate. Then Yersinia can easily be spotted with its unique characteristics. So Yersinia grow as a gray-white translucent colony, usually too small to be seen. Individual colony can be so small after 24 hours. So the colony have to be allowed to grow a little bit longer, where it can have more prominent features that shows the fried egg appearance. Yersinia is one out of all organisms in the family of Enterobacteriaceae, although it is not classified as Yersiniaceae. So it has these unique features of fried egg appearance, and this becomes more prominent as the culture ages. There are also oxidase are urate negative and indo negative and catalase positive as the previous classification under the family Enterobacteriaceae, although now in its own different family. In terms of economic importance, they are responsible for human disease such as plague and Yersinosis, and uncompleted cases of Yersinia will be due to enterotica, which may resolve on its own or with the use with or without the use of antibiotic treatment. But in the case of complicated cases, severe cases of Yersinia infection, treatment with antibiotics may be required, such as aminoglycosides, fluoroquinolones may be useful for the treatment of Yersinia infection. That's all for the study today about this wonderful member of bacteria belonging to Enterobacteriaceae and Yersiniaceae. Like I said, Yersinia used to be under the family Enterobacteriaceae. So we can still study together as a member of Enterobacteriaceae, but it has been reclassified. So with advances in more differentiation, genomic studies, we will be able to look at some other species that will come up under this new family. So I will have some questions for us to look at after this.